Good morning, everyone, and it's a great honor for me to be presenting here. Today, I'll be presenting our work on something which is very close to my heart, on trying to generate corneal endothelial cells in an ex vivo model. So none of us need to be told why we are doing transplants. Obviously, there's always a corneal need, and most commonly for some conditions like this, like Fuchs endothelial dystrophy, the treatment is endothelial keratoplasty, or in more advanced stages, we are looking at doing a full thickness corneal transplant. But we all know the severity of global shortage of tissue and, of course, the problems related to corneal grafts which lead to a graft failure and thereby worsen the vicious cycle of loss of tissue or shortage of tissue. And we're looking at ways to bypass this in whatever way we can. And one of the most tried methods around the world is to try and generate and transplant corneal endothelial cells. And of course, we are aware of the landmark work which has been going on by Professor Kinoshita and group, where they are trying to generate corneal endothelial cells from other corneal tissue. The challenges there is, of course, we are using an already limited tissue to try and generate more cells. And because it is from a corneal endothelial tissue itself, we know endothelial cells don't regenerate. They stop at G1 phase. So we have to reverse it and then move forward again. And because they are originally corneal endothelium, it tends to go into fibrosis much more, which is the endothelial mesenchymal transition, and the cells die out much easier. Also, there's a problem with transfer and viability. So the plan for our work was to try and generate induced corneal endothelial cells from peripheral blood, which we have ample of, and then move into integrating this into the substrate and make sure that it maintains the morphology and function, look at what alternate approaches we have, move into animal experiments, and then go to clinical trials. So our work till now is that we have taken peripheral blood, normal blood sample, a peripheral blood, which was reprogrammed. Those mononuclear cells were reprogrammed to form pluripotent stem cells. And as we know, stem cells can be then moved into any form of a type of cell, and then we moved it using, through the neural crest cells into a corneal endothelial type of cell using the Sendai virus. These were then characterized because we have to look at their clinical characteristics and also different markers which are known to be specific to corneal endothelium using RT-PCR and immunohistocytochemistry. So it may not be very visible. If you look at the third slide here, third image here on the left, the cells have over time formed the normal hexagonal pattern which we are used to seeing for our endothelial cell on specular microscopy. And these last for about 1 month or so, and these were derived from peripheral mononuclear cells from our peripheral blood. We then moved to see the markers. Zonular occludence is a very, very important marker for endothelium, which is a paracellular barrier and regulates the permeability of the endothelial cells. We all know about the sodium-potassium ATPase pump, which is responsible for dehydrating the cornea, and the non-muscle myosin, which is an essential process for migration of the cells. So all of these markers were found to be positive in the cells which we generated in the corneal endothelium. So this was done, and then we moved on to the next step of our plan, which was the integration. So the way we did it was get the institutional ethics committee. We looked at 10 cadaveric tissue eyeballs, and by a standardized protocol, we then looked at how to integrate these cells. So on the right side, the video playing is how we do atraumatic denudation of the endothelium. You have to make sure, because we need the DM there to remain there as a substrate for the cells to grow on, it has to be atraumatic, and we cannot strip off the DM how we do for a normal endothelial keratoplasty. The cells were injected in two ways. One was into an intact eyeball, and the other was placed onto the corneal button to see which would adhere better. And then after a period of time, we looked at the marking onto the cells. These are the specular microscopy images of these cells before endothelial cell removal. After endothelial cell removal, you can see no cells visible there. And after the cell injection, you can see that the cells have started adhering. They will be visible only if they adhere. Otherwise, it would not be visible. Similarly, the same markers we looked at earlier, the ZO1, was found to be absent in the decellularized donor cornea and again present once we had injected the cells and incubated the eye. Very interestingly, even the functionality of the cells was proven by the sodium-potassium ATPase presence and the non-muscle myosin. So it shows that these cells not only look like endothelium, they are also already functioning like endothelium after integrating onto the cornea, the cadaveric cornea, by injecting the cells. So where we are moving forward now is looking at other ways of doing tissue injection or delivery. The various scaffolds which we can use is like trying to form a DMX scroll. So using a DM itself or using amniotic membrane. And this is where we are at. We are now moving to our animal experiments, which is going to be a New Zealand white rabbit. And the choice of that animal is because the corneal structure and morphology is very similar to human beings. So this is where we're at. Thank you for a kind listening.
Thank you. Sharon, you had a slight error in one of your slides where it, about 5th or 6th from the last, where you showed before, and it should, the middle slide should have been after removal, but both slides, both the pictures had before, yeah. So just get that changed if you're going further up. Yeah. Yeah. Any questions?
I think wonderful study.
After how many days you did the injection?
Sorry, ma'am?
After how many days of removal you did the injection?
The injection is done immediately, ma'am. So we remove, it's an eyeball. So in the OT, we do the denudation. We have to wash out the cells which we have denuded now so that they are not residual and re-adhere. Then once that is done, then we do the injection. I sutured up that incision site to prevent leakage and then inject it from another site using a 33 gauge needle, 20 lakh cells. And then we do it immediately and then we incubate it for 3 weeks.
And the rest of the experiments after 3 weeks?
Yes, so after 3 weeks, we leave it in the organ culture media. Then we will harvest the cornea in the globe and study it again.
Thank you. Thank you, Sharon.